Takei. I'm from Toho University, Japan. First of all, I'd like to thank you for giving me this opportunity to talk my recent study. And what I want to talk today is that CXCL1 induced expression of BDNF. It is a famous neurotrophic factor. Before showing our results, I'd like to have a small introduction. Generally speaking, time is fair to everyone, but in some cases, maybe it is not true. This is an example. And the authors of this manuscript measured progression of biological age for 20 years. What they found is that most of people get one biological age per year, like this. But some of people get 2 biological age per year, but others get 0.5 biological age per year. So the authors indicated that progression of biological age is not fair. What is the decision factor for progression of biological age? Strictly saying, we don't know that. But what we know is stress and obesity promote the progression, but exercise inhibits or reverses the progression. So lifestyle can be the decision factor for progression of biological age, but we don't know the mechanism at all. What we found in PubMed is that exercise induces expression of a chemokine, CXCL1. But the role of the CXCL1 in the exercise-induced inhibition of biological aging is just unknown. So in this study, we asked whether CXCL1 can affect biological aging or not. To ask the question, the CXCL1 or saline is injected into the peritoneal cavity of aged mice. These plots are a typical result of flow cytometric analysis of peritoneal cells. And horizontal axis is forward sculptured light, and vertical axis is F480 macrophage marker. And compared with young mice, the peritoneal cells in aged mice contains reduced proportion of macrophages and increased proportion of lymphocytes like this. And CXCL1 administration had no effect on the age-associated changes of the peritoneal cell population. Next, we asked the phenotype of peritoneal macrophages. Phagocytic activity of peritoneal cells is known to decrease with advancing age. So we asked it. To ask the phagocytic activity, latex piece coated with a rabbit IgG FYTC complex is injected into the peritoneal cavity of aged mice. About 10% of peritoneal cells incorporated in the aged normal control mice, like that. But after CXCL1 injected, about 20% of cells incorporated the beads. So this result indicated CXCL1 promoted the peritoneal phagocytic activity in aged mice. And the cyclin-dependent kinase inhibitor of P16INK4A is a famous established marker for senescent cells. And vertical axis is P16 expression, and horizontal axis is P480 macrophage marker expression. So P16 positive macrophages in young mice is less than 5% around. But P16 positive macrophage in aged mice is 50% around. Injection of CXCCL1 reduces the proportion of P16 positive macrophages. So P16 positive macrophages was accumulated in the peritoneal cavity of aged mice, and CXCCL1 restored it at least partially. Taken together with the previous slide, CXCCL1 improved age-associated phenotypic changes of peritoneal macrophages. Next, we asked the effect of CXCCL1 on cognitive aging. In this experiment, mice explored this open field containing with two identical objects. 6 hours later, one object is changed to novel object, and again, mice explored this open field. If mice forget the familiar object in these 6 hours, mice will show similar interest to both objects. If you remember, because of their seeking behavior, mice spent longer time for the novel object. Here is the typical behavior of saline-treated mice. Enough, maybe. Oh, next, this is a CX3CL1 treated mice. I hope you can see the difference. Anyway, this graph summarizes the results of novel object recognition test. Young mice, about 70% of total explosion time spent with a novel object, but aged mice, just 50%. That means aged mice forget the familiar object. But after treatment with CXCCL1, the score is improved like this. This graph is from female aged mice. Again, CXCCL1 treatment increases the novel object recognition score. So these results indicated CXCCL1 restored the recognition memory impaired with advancing age. So next, we asked how CXCCL1 in the peritoneal cavity affects the brain. We found BDNF expression in the hippocampus was increased after the CXCCL1 treatment. The images were slices of hippocampus. Red indicates the nucleus, and green indicates BDNF. After CXCCL1 treatment, BDNF positive cells are increased like this. And vagotomy, that is excision of vagus nerve trunks, inhibit or abolish the increase of BDNF expressed cells like this. So these results indicated CXCCL1 augmented BDNF expression in the hippocampus, and the vagus nerve is the signaling pathway between the peritoneal cavity and the brain. What is a BDNF? BDNF supports survival of existing neurons and promote neuronal plasticity. And moreover, it is essential for maintaining adult neurogenesis. What is adult neurogenesis? It can be observed throughout adulthood in two regions. One is subventricular zone, lateral